Hi guys, welcome back to another video here. This is going to be a short one just going over the synthesis of biological macromolecules, those carbs, lipids, proteins, nucleic acids we talk about. How do we put them together? How do we break them down? Obviously that's important. You need to be able to put things together and you need to be able to break them down. And so we're going to talk about how we do that. So our first reaction we'll talk about is dehydration synthesis. So using context clues, just looking at the name, dehydration means a loss of water. Synthesis means to put things together. And so this is going to be an anabolic reaction. Anabolic meaning to build or put things together. Okay. And another name for this reaction, in biology we actually can call this another name, is called a condensation reaction. Still, condensation, we're talking about forming water. And the point of this reaction is that you are going to build molecules or put things together with the loss of water. Okay, so dehydration, we lost water. Synthesis, we're putting things together. So if you look down at our image here at the bottom, we have just, carbon rings. And these would be carbohydrates, like maybe a glucose. Okay, and when you look at the structure, and if you notice as you go through, whether you're in a bio class or if you've already taken one and you go back and think about it, or if you watch my videos as you go through, if you notice on a lot of these structures for these biological macromolecules, okay, if you look at, say, carbohydrates, proteins, lipids, if you notice a lot of times on the end of the molecules, where, the ends are going to be the ones that maybe react with something nearby, a lot of the times you see a lot of hydrogen and a lot of oxygen, either hydroxyl groups or just a hydrogen sitting there. And that's for a reason, because it allows this reaction to happen. So if you notice down here, we have this hydroxyl group highlighted in red, and we have this hydrogen highlighted in red. This is where water is going to leave. So H2O, OH, and H, this will leave as water. And so we're going to form water as we build something. Now, when we remove that water, okay, we are now left with an opening there. These molecules are now left with an opening. They need a bond to fill. And so what do? We actually can form a bond. And so in this case, this is a carbohydrate. This would be a glycosidic linkage. That's just, the name of the bond. But what's really important is that we were able to form a bond by removing water and allowing a new bond to be formed. Now, another, kind of terminology just to be familiar with, the little building blocks that we're using, okay, the building blocks we are using to put together, or the pieces that we're using to build the bigger pieces, these are called monomers. Okay, monomers are the small pieces, the building blocks, if you will, and as we start adding them together, we start to form polymers, or larger pieces, okay? And so through dehydration synthesis, or a condensation reaction, the removal of water, okay, so we remove water, we form that also. We remove water, and it allows us to form a linkage and bond these two monomers together to start building a bigger piece, a polymer, okay? So then our other reaction, the flip side of this, is going to be hydrolysis. Now, if you look at the name, hydro means obviously water, and lysis means actually to split. So you are adding water and you are splitting it apart. And so if you take a look at the image right off the bat, you should know or notice that this is the exact opposite of what we just did. So now we are adding water in, and we're going to add it back into that place where we just took it out, and we're going to separate that bond, and now we go back to what we started with, a hydroxyl and a hydrogen back in that spot. So we can break down polymers by doing hydrolysis. We can break them down into the smaller pieces, the monomers, the building blocks, okay? This is going to be a catabolic reaction. Catabolic means to break down, okay? Now, the last key point I want to note with this, and again, I told you this would be a short video, so pretty easy concepts here, just looking at what's being removed and how we either build or break down. What we have to know, or what we should keep in mind, is that this is all sped up by enzymes. Enzymes are proteins that speed up chemical reactions. So these enzymes are going to help with the building of a molecule, or the breakdown of a molecule is going to help with the removal of water, or help water get put back into the molecules, okay? But the enzymes, there's no specific hydrolysis enzyme and no specific dehydration synthesis enzyme. But these enzymes that function in either the building or breaking down of certain molecules will help these processes happen. Okay. For example, with carbohydrates, if you're trying to break down a starch molecule, so breaking down will be a hydrolysis reaction, there are enzymes, for example, like amylase. Amylase is a starch breakdown enzyme. It helps the breakdown of starch, amylose, but it does that through hydrolyzing. So that enzyme is not only going to help the breakdown, but it's going to help the hydrolysis piece happen. And that is really how you build molecules and how you break them down. So I hope that was super helpful, and I'll see you in the next one.